Good afternoon, everybody. My name is Jure, and I'm the account representative for the LabChip at PerkinElmer. Thank you for joining me for this webinar about the LabChip. I'm glad that there's so much excitement around the technology, and I look forward to kind of disseminating some of the uses and explaining how it will be good for your workflow. So first thing we're going to go over are the basics of microfluidics, the specific GXII platform, and then our assay portfolio. All right, so the first thing to know is that PerkinElmer is a microfluidics company. So we've been manufacturing microfluidic chips for decades at this point. We manufacture the chips for Agilent. We actually manufacture the chips for BioVenture, and we actually manufacture our own microfluidic chips. You can see at the top right of the screen. So the blue is for nucleic acid, the purple is for protein. Green used to be for RNA, but it's now grouped in with the blue, which does all nucleic acid. And we also have multi-sipper chips, which work on our sister platform, which is a derivative of the LabChip. So you've been around the block quite a while for microfluidic chip technology. So we're quite confident in our ability to manufacture and perform these analyses. Specifically for the LabChip, what you need to know is that all of our chips are made with a high precision photolithography process. So you can see this micrograph at the top left. So there's a precise channel geometry defining the fluidic circuit that we use for analysis. So all of our chips are made of quartz. There's two parts to the flow that I'd like to, to the analysis. There's electrokinetic flow, and then there's vacuum flow, which is going to make up kind of the total electroosmotic flow. There's one excitation, one emission, the 635 and 700 nanometers respectively. And then what a lot of users love about this technology is that it's quote unquote smart technology. So you could run 100 samples on a LabChip in San Carlos or where have you, send that LabChip to any other part of the world. So send it to Boston and then put it on a LabChip unit that's there. And it will tell you that that microchip is deficit 100 samples. So it's always going to tell you what has been run on the chip and how much is left on there. So it's always reading from and writing to the chip. So it's a really neat thing to know about the system. So at this point, I actually like to show a little video that demonstrates what's happening on the technology. So what I'm going to do is, let me see if I could do this in a nice kosher way. I'm going to see if I could switch over to the screen here that I have this video. Right, could someone confirm that they could see this other screen before I continue?
Yes, we can see.
Okay, fantastic. All right. So this is a LabChip system. It's 20 by 20 inches and it'll fit on any lab bench. I actually put in my Subaru. And our tagline is, why do gels? So that's the technology that we're replacing a lot of the time. So how does a LabChip work? Well, let me show you, it's quite easy. So it's a microchip, or excuse me, microtiter plate-based system. So it works with 96 and 34 well plates, so high throughput plates. It is cartridge-based as well. So the microchips are reusable, which is a nice thing about our platform over other platforms. So if we take a look inside where the magic happens, there's a robotic stage that's going to move the plate cartridge under the zipper of the chip. All the plates that we use within the system have a specific XYZ geometry that allow us to perfectly position the zipper of the chip in the bottom center of the well when we sip up sample. When we begin analysis, we're first going to apply negative pressure at a vacuum well, and this is going to allow us to sip up some sample. When we sip up sample, at this point, we're only sipping nanoliters of your sample. Once sample is introduced on chip, sample is then going to mix in a 1-to-1 ratio with our first internal standard, which is our marker solution. And that's going to be it for the pressure-driven part of the analysis. We're next going to apply voltage between wells 3 and 8 to inject sample to the right, and then between wells 7 and 10 to inject sample up. So this is a cross-injection platform. Your receiving matrix is primed throughout the entire capillary. So we're only going to inject a tiny plug and picoliter volume that's going to get separated by size in the capillary that has sieving matrix. The next thing that we're going to do is apply D-stain for protein analysis. So we're gonna apply unlabeled SDS to drop the SDS below the critical micelle concentration so that we're able to detect the protein against the background of the baseline. So that's what's happening between labeled and unlabeled. The next thing is we're going to excite with a laser. So it's a laser-induced fluorescence platform. So within the same region where we're exciting, there's actually a detector. So what you'd expect to see on the front of your instrument in real time is an electrophoregram that's generated second by second. It's going to show you each of your peaks and all your data. So one thing I'll go back and mention that's not quite clear here is that this robotic stage that's moving under the sipper, the robotic stage in between each injection will actually always go to a wash tube to make sure that there's no cross-contamination between samples. So we only take the ride through the fluidic circuit for this one sample within this video, but it's important to understand that the actual sample list is going to be such that it's injection wash, injection wash, injection wash. So does that make sense? I think it does make sense. But if if you guys have any questions, feel free to chime in as well. So we apply negative pressure to the vacuum well. We sip up sample, mixes in a 1-to-1 ratio with the first internal standard. We apply voltage between wells 3 and 8 and between 7 and 10. We inject into the capillary separation by size in the sealing matrix. We excite and detect within that same region. You'll get an electrophoregram within 42 seconds at least. All right, system and software. So everything that you need to complete analysis, we provide for you. It's a 4-component system. It's instrument, software, chips, and reagents. And then there's an optional 21 CFAR Part 11 compliance package for users that eventually end up going into manufacturing. And I'll speak a little bit about that as well. So this is the control software called the LabChip GXII software. So to orient you to the system a little bit more, one thing that I think is a glaring, really cool thing about the system that's obvious is that it has an onboard computer. So I always describe it as being a computer superimposed on a high precision bioanalytical instrument. So if I were to press unload chip, the chip door would open as you can see at the bottom right. If I were to press load plate, plate cartridge would load back into the instrument off of the left-hand shoulders you could see there. Prime, run, and wash are all functions that you're able to do in the lab when you're running analysis. But the nice thing about this touchscreen platform is that, and this is more of an upgrade from the predecessor to LabChip, is that it actually has increased ease of use dramatically. So compared to other platforms, we may need to click through complicated menus to set up your run, adjust voltages and pressures and what have you. This touchscreen format has made it easy such that you're able to easily kind of click and get to where you need to go to perform the analysis and identify all the parameters that you need in a more succinct way that's more intuitive. So at this point, when we do trainings for LabChip, it ends up being, one day for some of our base assays. So ease of use dramatically increased with the touchscreen. The other nice thing about the software, at least the control software, is that you're able to get a quick look at the data a priori without even having to go into data analysis mode. So say that you're running a 96 well plate or 384 well plate, you do not have to wait for the entire plate to finish for your data to start becoming available to you. So say that you're running one of these plates, the first injection that will come off is your ladder. The second that ladder is available, you're able to go look at it in views and make real-time decisions about your samples as they come off injection after injection as well. So this is a huge time saver to a lot of people, especially if you are running, a lot of samples at once. Say you're looking at clones, you don't need to maybe run the entire plate if you know that the first 2 columns are passing, what have you. So it is a really nice ability to get that real-time feedback from seeing the electrophoregram generated in front of you, having that data available to you in the control software without having to actually, click into another software or wait for an entire plate to finish before you realize that you didn't really have to do all that analysis. So it's a really powerful ability. And in this case, it's a lot of time saving. All right, so this is the data analysis software called LabChip GX Reviewer. So what I love about the software is that it's amenable to any throughput that you'd see feasible for you in your laboratory. So to orient you a little bit, this is a laser-induced fluorescence platform. And with that, you get a nice electrophoregram or chromatogram and electrophoregram form. So, but with that, if you still like looking at gels, you know, living in the past a little bit, we still give you that option to visualize your peaks or your moieties in the gel format. So the gel view, which you could see to the right, it's great for comparative analysis of a lot of samples side by side. So if you needed to maybe compare bands and see if one band shows up in one sample, not another, we give you that view as well. So all of your quantitative information is actually going to show up in the table at the bottom, in the well table and the peak table. So it'll be a more of a tabular visualization. So we have two ways of visualizing data. There's the well table and the peak table as far as for the numbers. So well table will usually give you a concentration data, well by well, and then peak table will allow you to parse data by specific peaks that may show up throughout the entire plate or any well on the plate, excuse me. So the reason I like this software a lot, and a lot of people will kind of speak to this as well, is that it's actually free. So there's no seat licenses. So you can go on the PerkinElmer website right now and actually download the software and do data analysis at your computer, as opposed to having to be in front of the instrument and doing it on that specific, you know, with your lab coat on, with a mask. So it is very nice to kind of have that ability to analyze the data anywhere that you go. So what you look at when you see that the top left is that there's a plate, you're actually able to open multiple plates and do analysis across different runs. So say that you want to compare an experiment that you did today to an experiment you did yesterday, it's an experiment you did last week. You're able to do all of that together, and the software will actually automatically do the analysis for you. So say that you're looking for 72 kilodalton moiety with a plus or minus 10% window of tolerance. You then tell the software, if you identify this moiety, please highlight that well with the color magenta, and it does so. And that makes it easy so that you don't have to click through each well and manually integrate and try to find out, you know, does that moiety exist? What's the concentration? What's the size? The software will automatically do that for you 99.99% of the time correctly. That isn't to say if you look at the baseline one day and you're like, oh, that's a little bit off, you can absolutely go and adjust it, but it's more so, you know, powerful for you because you don't have to do the manual clicking. So that is to say, if you wanted to apply the settings, I'm looking for the 72 kilodalton moiety to 1 well, to 100 wells, or to 200 wells, it's going to take you about the same amount of time. So how it works is you say, these are the parameters that I like to search for. I want to apply to these specific wells. And that's what makes it really easy for you to do analysis across any throughput needs that you or your group has kind of in store for your work. So it's really nice to be able to use this function. And as I mentioned, it's free and there's no C licenses. And then there's an optional 21 CFR Part 11 compliance package. So the LabChip is heavily used in biopharma, as you guys would expect. It's brought about 20 drugs to market. So within the GMP or the GXP software, there's a central data repository that allows all of the data to live on the computer in the kind of a lock and key area that you're able to set access levels for each user. So there's certain privileges where maybe a director level person may be able to access this data and do some copying or analysis, where a manager and, you know, someone that may be just a regular scientist may not have that level of privilege. So it makes it really good for kind of audit trailing and auditability. So if the FDA came knocking on your door and they're like, show me your batch records, all of this is kind of automatically set up for you in a nice, neat little kind of bow package that's ready. So we could talk about this at length. If you guys have questions, feel free to reach out. protein applications. All right, so if I've kind of alluded to, the system's quite versatile in its ability to perform analysis. So what I think is really important to kind of highlight as well is that in addition to protein analysis, which I've kind of shown you guys some slides on already, it's able to do analysis for DNA and RNA. So it has a solution for all of dogma as far as biophysical characterization goes. So for this particular slide, it is protein focused. So I will talk about what a lot of our protein kind of clients like to use it for. So that may be looking at concentration, size, or purity, or visualizing aggregation. You are able to look at stability and look at a degradation of your sort of products if you were in a manufacturing setting. And then we actually have some neat assays where you're able to do end-link glycan profiling. You're able to do charge heterogeneity. So look at the charge on your monoclonal antibodies. And then, as I mentioned, it's used kind of heavily in QA/QC release. And then reduce and non-reduce is no problem for the system as well. So with that, I'm going to go into the assay portfolio. I'm going to speak through it a little bit. So starting off with our workhorse assay for protein, which is Protein Express. So Protein Express is the most popular assay for the LabChip. So it's been on the market about 2 decades. It's one of the first assays that were developed for the system. It reports you concentration, size, and purity, and has a molecular weight range from 14 to 200 kilodaltons. It is 42 seconds per injection and is 400 injections per chip. So I mentioned it's brought about 20 drugs to market, extremely robust. It is, I'd say, the one that most people I'd recommend start with. But say that you needed a little bit more resolution specifically on the lower end, maybe the 5 to 80 kilodalton range, we have a low molecular weight Protein Express assay. So this particular reagent kit is going to give you the same analytical output, so concentration, size, and purity, but it's going to be 60 seconds per injection. But, I don't think you're going to lose any sleep over that. And it's 400 injections per chip as well. So I'm going to speak to how Protein Express and low molecular weight assay work first. So these assays work via staining method. So we're staining the protein with SDS that's been labeled with dye. And then due to the amphiphilic nature of SDS, it causes the micelle to be formed. Then you remember the kind of the rigmarole. We introduced some unlabeled SDS, dropped the SDS below the critical micelle concentration so that we're able to detect the protein against the background of the baseline, excite, detect, and then there we are. So Pico Protein Express is a different flavor of assay, and I'd say it's so this is actually the second most popular assay for the LabChip. And this was the first assay that was developed, actually. So this is the most sensitive assay in our portfolio. It affords picogram sensitivity, and we actually directly label the protein via the lysine. So we actually do conjugation of dye to the lysine of the protein. And this assay would work kind of similar to how traditional chromatography methods, for those of you that may be familiar with them, are kind of set out. So you'd optimize your dye to protein ratio. You'd optimize your heating steps, so how long you're heating for and what temperature you're heating at, to find out what conditions are going to give you phenomenal labeling efficiency. So once you've kind of optimized that method a little bit, you're good to go. So Pico Protein Express works very well, especially if you need that sensitivity and you're working with kind of very dilute species. So I'd say as far as popularity, Protein Express is the most popular because of the ease of use, which I'm going to talk about in a little bit. But Pico Protein Express is right there behind it as far as what it's able to give for you. You're able to look at aggregation and then the sensitivity that it affords and kind of all of the benefits we talked about. So then we have a glycan profiler assay. So the nice thing about this assay, which is pretty neat, is that instead of having to send your glycan samples to a core lab, say that you actually just wanted to determine your glucose content, your mannose content, so, your G0s, your MAN5s, what have you. Our assay works such that for your glycans, we denature, we digest with PNGSF, we then conjugate dye to them, and then we run them on the LabChip alongside standards. So usually you'll have standards you can get from QA Bio, you can get from Prozyme, and then you'd run those standards alongside your samples on the system, knowing what the concentration, the input concentration that you're introducing is, And then based off the area under the curve, you'll be able to come up with a standard curve that allows you to back calculate what the concentration is of those glycans on your monoclonal antibodies. So you're able to reliably get glucose content, mannose content, what have you, in a matter of seconds, as opposed to the kind of the lead time that it may take to send this out to a core laboratory and do some mass spec. All right, so then there's a charge variant assay. So the charge variant assay allows you to look at the acidic and basic moieties on your monoclonal antibodies. So this is, I want to say, an orthogonal method to capillary isoelectric focusing. So capillary zone electrophoresis, and how it looks like on our platform is that we actually have two assays that are going to be dependent on the PI range of the protein that you're looking at. So we have a low PI assay that goes from about 3.5 to 7. We just released that one. We have a high PI assay that's going to go from 7 to 9.5. So how that assay works is within the kits or the reagent kits for this, we actually provide you pH standards. Those pH standards you're then going to use to make a running buffer of a certain pH relative to your PI of the protein. You are going to have it be maybe a unit or two above or below, and then do some method development to determine what pH is going to give you the best separation. So you may try a running buffer of 7.1 when your PI may be 6.5 and see how it turns out. And if that you can get a little bit more a resolution or separation, you may try 7.15 or 7.2. I'd say the strength about this platform over CIEF is that it's a lot quicker than CIEF. So we give you kind of 3 running modalities. You can run for 60 seconds, 90 seconds, or 110 seconds. You know that it only requires 60 seconds to get optimal separation. You're able to choose that one as opposed to using something that may take you a lot longer per actual sample, like a CIEF. All right. And then I'd like to mention that within this system, this is more of a, this makes more sense in comparison to the predecessor to the LabChip, which is the blue box. There's technology inside of here that actually allows us to run some neat calibrations that make sure that we afford excellent coefficient of variation of a really tight CVs, so a really low coefficient of variation. So with that, intelligent technology is a hardware in the system that allows us to optimize the separation voltages, optimize the destain currents, optimize the laser power, so that every time that we measure a standard, and for these particular assays I'm willing to discuss, we actually source our standard from NIST, so that we use NISTmab, we use Verimab. So every time that we measure these calibrant standards that we're going to use, and we can measure the area under the curve, it's going to be of negligible difference. So you may see 0.0001 percent difference from the times that this standard may be measured. So with that, that's intelligent technology, and we have an advanced assay portfolio that includes Protein Clear and Protein Exact that uses this technology. And then for comparison, I have Protein Express to the left. So first starting with Protein Clear, since it was the first assay. So this is our purity assay. This is the assay that our pharmaceutical clients and partners are using when they're going for QA/QC release sort of measurements. So Protein Clear uses a two-step calibration. So it calibrates using Verimab, the standard we have from this. It also calibrates using our ladder standard. This is going to use a high resolution chip. So this high resolution chip actually allows the quantification of a broader range of proteins. So we have a 14 to 250 kilodalton range for this assay. This assay will only report purity. And then as far as reproducibility, it has excellent intra-run and inter-run reproducibility. So less than 0.5 on our coefficient variation on our main non-reduced IgG, which are 120 kilodalton peak within our ladder, and less than 5% on all other peaks. So then Protein Exact is the next one that's up. I like to explain it as being a marriage between Protein Express and Protein Clear. So Protein Exact is a single-step calibration. It will calibrate using our ladder standard. And then this is a high-resolution chip as well. Protein Exact currently has the broadest molecular weight range of any assay we currently offer that goes from 6.5 to 250 kilodaltons. So this assay will still report to you size and concentration, addition of purity, if that is something that was important to you. So if you needed kind of the best of both worlds, Protein Exact is very nice. So as far as reproducibility, we guarantee you less than 10% coefficient of variation on quantitation. And a lot of the applications that Protein Exact is being used for right now, I'd say an exciting one is gene therapy. So quantitation of AAVs. So the ability to kind of look at your VP3, your VP2 to VP1 and tell with good fidelity that you have a nice 10-to-1 ratio. So it is being heavily used in that aspect or in that regard. And we have application notes that are kind of reflecting that. So it's a really neat and I'd say an application that we're excited about for Protein Exact. So sometimes I get the question, Dre, why is Protein Express 30%? So the truth is, it is never 30%. It is never, ever going to be that high. This was a number that was chosen 2 decades ago when Protein Express was released. So I just want to make sure I get it on record. If it's ever that bad, you guys call me up, yell at me, I'll come and do the pipetting myself. But it is just something that we've reported historically. So it's important that you guys understand that you are able to get probably closer to 12 to 15 on Protein Express. All right, now we get to look at some data. So here, and the take home of this slide is going to be resolution, as you can see. So we're looking at a fermented page ruler and stain protein ladder in lane 1. We're looking at a fermented and stain molecular weight marker in lane 2. And the mixture of those is going to be lane 3. So the point here is that, looking at a regular page gel, if you're using silver stain, densitometry, what have you, you will not get as good a resolution, and we will definitely not be able to tell the difference between something that, is migrating at 26.84, 28.5 on, or has a retention time, or what have you. So the nice thing about using a capillary electrophoresis system and laser-induced fluorescence is that you do get phenomenal resolution. And that's something that you'll see a bit more of examples of as we go into the presentation some more. Sensitivity. So here we're looking at a dilution series of carbonic anhydrase. So from 2,000 nanograms per microliter down to 1.9 nanograms per microliter. The right image is an inset of the left image. So as we go to 15.6, 7.8 to 1.9 nanograms per microliter, you're able to make out that curve quite nicely. So the profile is still there. So we have phenomenal sensitivity as you go lower and lower and lower still. And this is just for Protein Express. As I mentioned, we have an extremely sensitive assay called Pico Protein Express. It's verified to go as low as 50 picograms per microliter. So if you need to work in that region, you're actually able to quite comfortably. That isn't to say take your 10 mg per ml sample and dilute it down to 50 picograms per microliter, right? It's more to say if you need to work in that region, will you actually help you with the method and assay development for that. So that may be, curtailing your surfactants and detergents or ionic strength, anything that that may end up impacting labeling efficiency will work with you to make sure that you're able to get the labeling efficiency that you need, but also still have kind of the buffer formulation that you need to maintain stability of your moiety or your sample. All right, and here we're just looking at an example of what the CESDS looks like for a monoclonal antibody. So the monoclonal antibody industry has been using CESDS for decades at this point. They love that it's quantitative, reproducible, it affords you digital records. So to be able to have that in 42 seconds, to do that kind of in a high throughput setting, it is nice to have that ability. And we're looking at this example for adalimumab. As I mentioned, we also do see applications with a gene therapy for adeno-associated virus. So users that may be doing sort of the bioreactor kind of design and the growing of these AAVs and being able to determine ultimately, once you've kind of worked through your process, have I purified my sample completely, you are able to easily visualize what contaminants may still be in the sample, as well as what's actually showing up. So do I see all of my viral capsids, or do I see any sort of indication that there may be something within this AAV? All right, Glycan profiling. So as I mentioned, the system is extremely versatile and we're able to do analysis for glycans as well. So here we're comparing the LabChip to a standard, CE system, let's call it competitor P. And then the point here is that on the LabChip, you're able to get the same results in about 160 times faster than using kind of standard capillary electrophoresis. And you're going to get the same sensitivity. You're going to get probably slightly better resolution as well. And then what we want to point out on this slide is that we actually have a new assay called Glycan extended range, where we're able to look at your salicylated species that are charged and will actually show up a little earlier. So if you do need to visualize those glycans that are charged, like your salicylates, this is something that you could do on the LabChip readily. And we do have application notes as well that I'll talk about towards the end. All right, so now we're looking at some charge variant data. So we're comparing the LabChip, which is microchip-based data in the top left, conventional CZE from competitor P, and then comparing it to an ICE 280. So the point here is that with the LabChip, in about 60 seconds, you get really good resolution of your basic, your main variant, and your acidic variants. Compared to competitor P, it'll take you about 5 to 6 minutes, and you're not really getting that good resolution. We are going to compare it to an ICE 280. You do get phenomenal resolution, and you are even able to make out the PI of those species, but it takes you 15 minutes still. So if we look at the kind of the measurement output at the top right, the point is on the LabChip, you're actually able to get equivalent or better data on the LabChip as opposed to the competitor technologies, and it'll be a lot faster using the LabChip. So the relative amount that you're seeing across all of them are comparable. All right, now we get to talk about some publications. So this is a design of experiment by Vertex Pharmaceutical in Boston, where they were using an E. coli expression system, and they needed to determine the optimal conditions for expression of their protein. So they had about 288 unique conditions that they needed to test. Do you imagine using kind of a competitive platform that doesn't have as fast turnaround, or using a SDS page? It would take you quite a while to do this sort of study. So this is something that the user were able to do in an afternoon using LabChip. So they praise it for making possible what if experiments that up to now had been either prohibitively expensive or too complex to perform. All right, so here's another example from the NIH, and they're using it for vaccine development. So a little bit of a relevant example here. So they're looking at 3 serotypes of encephalitis, and they're using VLP virus-like particles for vaccine development. So they're looking at Western, Eastern, and Venezuelan, and those are the 3 serotypes you could see in the image at the top right, fluorescence versus size. And then the bottom image is going to be area response versus VEE, VLP concentration in microgram per milliliter. So these authors praise the system for doing a similar thing, but they more specifically talk about kind of the sample input benefits. So the LabChip does not require that much sample input when you're doing development of these studies. No, it's only 2 microliters or less. So with that, they say this method improves sample testing of lower concentration vaccine candidates, both in process development sample and the final drug substance. So after the development phase, when they get to kind of the quality assurance phase, this also has a really good, I want to say, neat application, and that it's able to do that very fast. So you're able to determine the purity and look at sort of the stability of your product at the end. So it's really nice in that regard as well. All right. So just to orient you to the consumables a little bit, every time that you're going to run analysis, you're always going to need a microchip, and you're going to need a reagent kit that has a chemistry. So the microchip you can see at the top right, the reagent kit is going to have these contents in it for a protein kit. For DNA, it will look a little bit different, but colors will be mostly the same. So the dye will be blue, sample buffer will be white, gel matrix will be red, The ladder will be yellow, the marker green, and the wash buffer purple. And then we also include consumable items like spinner filters, what have you, which you'll need when you're running the actual protocol. All right, so this is a workflow. I like to think of it as kind of two steps that you're performing in parallel. So sample prep and chip prep. So sample prep is going to start with your 2 microliters of sample that you're going to add to the microtiter plate. So a specific well of a microtiter plate. And then you're going to add 7 microliters of sample buffer. Sample buffer could be of reducing or non-reducing nature. So you could use DTT, beta-mercaptoethanol, T-step, what have you. If you wanted to use a non-reducing buffer, you're able to use, an alkylating agent. So say you wanted to use IAM, iodosetamide, to make sure that, your sample maintains the fidelity that it needs to for analysis. So you're then going to heat that 9 microliter mixture for whatever you normally do for denaturing your proteins. So I've used it to do 70 C for 10 minutes, or you could do 100 C for 5 minutes, what have you. Usually while that's on the heat block, I would do the chip prep part. So what that would be is, if you remember, I take my blue cap dye, my red cap gel, mix it together, spin it down on microcentrifuge, and get rid of any aggregates that way. Then I pipette those reagents onto the chip. Once that's done, the thermal cycler or heat block will usually be done. I'll add 35 microliters of water to each well, do a quick pipette mix, and I'll pop everything on the instrument. Within 75 minutes, I'll get 96 injections that are complete. So it's these 42 seconds per injection within 75 minutes. With the heating and warming and priming steps, I then have all of the data for 96 wells. So extremely robust, extremely quick. So this is a buffer compatibility table, and this is a neat thing that we include in all of our user manuals. And users love this because it specifically allows them to kind of help themselves. Say that you're running analysis one day and you're like, I wonder why my sensitivity is off. So you're able to come to this buffer compatibility table, have a look at it and say, maybe my percentage of tween 20 is too high. Or maybe because I have tween 20 in the presence of 8x PBS when I know that my tween 20s is a concentration limit too. Which brings me to the important point that all of these buffers are compatible. The only incompatible buffer is RIPA buffer, but there's, at this point in science, there's other solutions for membrane solubilization. But this is to say, you wouldn't expect the ability to use 0.8% tween 20 in the presence of 8x PBS. So these are for all those reagents in isolation, but use that information when you're kind of developing your method and determining what trade-offs are you willing to have within the buffer formulation when it comes to getting the efficiency or the labeling efficiency that you need for detection. All right, nucleic acid analysis. All right, so as I mentioned, the system is very versatile, is able to do analysis for nucleic acid. Works very similar. So 635 and 700 nanometers respectively. And then applications that may be of use to some of you guys, maybe PCR analysis, genomic DNA QA/QC, spare analysis for NGS. So we may touch on some of those as we go through it. But the point here is that the workflow for DNA analysis is actually quite similar. So it's actually easier than protein analysis. So you'd simply dilute your DNA to the concentration for linear quantitation of that particular DNA within the assay that you're using. So simply looking at the specification sheet and diluting it to the concentration that's needed. And then for the chip prep, same thing. You take your blue cap dye, red cap gel, mix together, spin it down in microcentrifuge to get your aggregates, pipette everything onto the chip, and then 75 minutes, 96 injections. So it works very similar, and then DNA is very stable, and so you're able to kind of rerun it. So I have users that may prepare a plate, they may freeze plates, they'll come back and run it. So you don't have to add any special buffers or anything to it. And this is our DNA assay portfolio. So it's actually named such that for the sizing, the size base pairs that it's able to size up to. So 1K will size up to 1,000, 3K to 3,000, and so on. So what I will I'll kind of highlight the same things that I did for the protein portfolio. Our workhorse assay is a DNA high sensitivity assay. So this was the first nucleic acid assay that was developed for LabChip. So Size range from 50 to 5,000 base pairs, concentration range from 10 to 500 picograms per microliter, 68 seconds per injection. These chips are actually 2,000 injections per chip, so you're going to get quite the mileage out of them. And as I mentioned, this is a workhorse. So as far as our most sensitive assay, that's going to be our DNA 3K assay, also termed NGS 3K. So I do have a lot of users that use the LabChip specifically in the diagnostic space that may be using it for a library QC. So if anyone does NGS, they do a library prep and they want to check at different points in the process for library prep. So even if you're starting at cDNA synthesis, you wanted to QC your RNA, or you had you're at some point where you wanted to see if you had a primer dimer, what have you. You are able to use the LabChip to easily do that. The 3K assay comes in for library QA/QC when you know that at the end of library prep, you may end up with about 20 microliters of sample sometimes. So the nice thing about this assay is that the input is going to be reduced compared to our other assays. So you only need about 3 microliters of sample for a 96 well plate if you're going to be using it for QCing your library. So that's huge because it leaves you all of that other material to actually put on the sequencer. All right, so same thing here, we're just gonna look at some data. So resolution on the LabChip as far as nucleic acid gels, you're actually able to make out something as far as a 413, 417, 427, triplet, a lot easier on the LabChip as opposed to looking at, another electronic gel and not being able to get that resolution very easily. And then speaking to the NGS application that we talked about, smears are easily analyzed on this platform. So when we do integration, it is more of a drag and drop sort of mechanism. So you are able to integrate just regions if you'd like, or we could integrate an entire sort of a well across the area under the curve. So integration within our platform, where we're looking at smear analysis, is quite easy. And then as you can see in the apply to wells sort of menu here, you could say I want to apply it to all wells or apply it to one well. So it makes it kind of quick and easy to do this analysis. So here we're just looking at some example ladders and some example data of what you may see as far as peaks on the lab. So these are particularly our ladders. So DNA 5K is an assay, DNA 12K is an assay. So if we were going to compare by looking at maybe we're running commercial standard on our assay. So if we're looking at DNA 5K and we're running this, I'm not going to even try to pronounce the name of this ladder, to be honest with you, but the point is, if we're running a commercial ladder, we see that what's actually measured is quite close to what the actual size is based off of what the manufacturer has reported. So it's quite close, and the same thing for DNA 12K. So measured versus expected size lines up pretty nicely. So if you are looking at other methods to maybe more high throughput methods to QC your nucleic acids and get concentration or size, it is quite easy to bridge other technologies to the LabChip. So I just want to make sure that was kind of clear and readily available for you guys to see. All right, and then we're able to do RNA analysis as well. We have a few different flavors of RNA assay. So we have a Pico RNA assay, we have a small RNA assay, we have a total RNA assay. So whatever your application, reach out to us, we'll be happy to support you on that. So I've had questions about the ability to get a RIN score on the LabChip. You are able to get a RIN score equivalent called RQS score, or RNA quality score. So these are going to take into account sort of what Agilent's method is using as well, the 28S peak, 18S peak, determining kind of the area of the curve and at what region that is migrated in to ultimately give you a RIN score, or which is going to be RQS score on our platform. All right. So here we're looking at some RNA analysis data, and we're looking at specifically 3 time points And the take home here is that we all know that RNA itself is not the most stable of molecules. So all of our hands, whenever we're working with RNA, you know that our hands have RNases all over it. So the point of this slide is that when we're looking at data that's been on or a sample that's been on the instrument for at these 3 different time points, the RNA has not degraded at all. So we are able to reliably come back and measure this, and we're looking at rat brain tool RNA. So the RNA is stable within our system on the platform. And then kind of wrapping up here, we're looking at the comparison between RIN and RQS. So you could see the RIN score for E. coli tool RNA is at the bottom, and then the RQS score that the LabChip gives you is comparable. So 5.8 to 6.0. Same thing for total RNA and wheat. So RIN score at the bottom to 7.4, and then RQS score smack dab on it, 7.4 as well. So it is another method that you could use and still bridge your data onto this platform. All right, so with that, it's been a pleasure kind of speaking with you guys through all the fun stuff you're able to do on the LabChip. I know that I have just been talking for a while. So at this point, I would like to ask if there's any questions, but before that, I do want to share this slide about your PerkinElmer Dream Team working at NBC Biolabs. So we're quite excited to work with you guys, and we're happy that you're showing sort of interest in our products. So we just like to remind you of the entire team, and across any of the platforms that you like, that sort of information about, you could reach out to us or any of these people. All right, so with that, I'm going to open it up for questions, and hopefully there's some more interest.
Hey, Dre, I think there's someone in the chat that had a message of based upon the software on the website. It's in the chat box.
Okay, in the chat box, might take me a moment to, let me see.
I can read it to you.
Okay. Oh, that'd be great.
Yeah, it says, so Pio, sorry if I'm mispronouncing, but it says, I went to the website to download the software, but the files are not available. Can you please double check? I clicked on LabChip GX Touch 1.10 under setup files.
Okay. Okay. I'm sorry about that. So what I'll do is I'll follow up. So if you're able to leave your email in the chat, I'd be happy to talk to you after and get you the software. I'm sorry about without the website. We'll figure that out if there's an issue there.
And I believe one other question that we've been receiving here at NBC Biolabs is how people can get trained to start using the instrument.
All right, and that's the best question, the most exciting question. All right, so we're still working this out. Training is going to look like at this point, we're going to designate days on the NBC calendar, and I will probably disseminate this information to everyone, but we'll designate days for specific assays. So the unique thing about the LabChip is that it has so many different assays that one training will not really suffice for us being trained on the LabChip. So we're going to probably do a couple trainings per quarter specific to an application. So one day may be CESDS, and then maybe in the following couple weeks or the next month, we may do a DNA training, and then in the same regard, a Glycan training and a charge variant training. So training will be more regular in that regard. If it comes down to specific users that are unable to make those trainings, that we'll call those office hours. you are able to reach out to us, and we'll see what we can do to accommodate you and get you personalized training. But that will be on a case-by-case basis. So generally, we'll stick to having scheduled trainings on the calendar. Thank you, Flavia. That's a great question, and thank you whoever asked it.
Jerry, what is the, sorry, somebody else was asking a question.
Oh, nope, I think it's all you.
Okay, what's the life of that laser that you have in the system? Is it certain readings, certain hours? Is there any issues with that LabChip, the laser that you've seen over years?
Nope. So I tell you that I've been in the R&D laboratory for this. So this is, I'd say quite literally my baby. I have not seen any issues with the lasers over, 7+ years. So it's nice that it is one excitation, one emission. It isn't such that there's something that you really have to change it out or do any sort of surgery. So it really is stable over time. We do have consumables that help us calibrate. So over time, if you needed to maybe perform an optics calibration with the laser, that is something that is included in a kit when the unit is delivered to you. So there's a calibration tool for the laser, but never a need to replace the laser. Does that make sense?
Yes, maybe it is something else because we had a system and there was some laser issues and that has to be fixed, and I was reverting to that question. That's okay.
All right, if you want to follow up afterwards, I'd be happy to speak about it, and I could also loop you in with anyone on our kind of internal team that could speak to it a little bit as well. Absolutely, if you'd want to speak more about it.
Hey, Jure, this is Serena. During office hours, the question that I get is like, where is the LabChip, and should they get trained by you before they start using it, or how do you want MBC researchers to interact with the machine right now?
Yeah, that's a great question. So right now, anyone that's walked past the LabChip will see that there's a big "Do Not Touch" sign on it, and that is because it is quite a finicky instrument, and it is very, it's a high precision instrument. So it isn't something such that we could train you virtually or, really just send you a manual or something like that. There's a lot more of a hands-on component to learning how to use a LabChip. So as of now, if you have not gone through any sort of hands-on training, you are not qualified to use LabChip quite yet. But that is something that we'll work to do and have everyone get trained up as soon as possible.
Yeah, that's wonderful. Great. Yeah, most of our partners, they do the same. So if you have different applications for the instrument and also for you to get to know the users, is we also suggest to have like a required training before using the instrument. And we have like our booking system, and before everyone get to be open to book the instrument, they will need to get trained, and once they get trained, you let us know and we will give the permission for people to start booking the instrument.
That sounds good. So and exactly. So I think the follow-up would be just getting users to register for those trainings when they become available. So I'll work with kind of our MBC Dream Team here to get that on the calendar, ideally before the end of this year. So you guys could plan accordingly for when you'll be available to come and go through those trainings.
And also, Jure, another question that I got during office hours is like, so everyone who was interested in using the LabChip, they would need to purchase the consumable first before they use the LabChip. Is that correct?
That's correct. So it would be ideal. I could do a bunch of hand waving, but without the consumables, we wouldn't get really far on using the LabChip. So We do need the users to have the consumables ready before they're able to perform analysis. So partly the nice thing about doing this webinar and why we push to have this is so that users can understand what assays are available to them and what applications they could use, as well as to get a feel for the different assays require different consumables as far as reagent kits and chips. So depending on your application, you will need to order unique consumables for that assay.
And just to let everybody know, we are working directly with our partner here, PerkinElmer. So if we have a lot of resident companies using the same consumables, we want to have it on site on our MBC shop. So let us know which consumables you will need, and we will make sure to have it on site as well. Any other questions? I know that we are quite there. Well, if nobody else has any questions, I would like to thank you once again, Dre, Serena, also. Thank you so much for your time, for all the continued support, and happy holidays.
Thank you, Flavia. Thank you, Dre.
Thank you so much, Flavia. Thank you, Serena, for organizing this. It's my pleasure. Everyone, have a fantastic holiday season. I appreciate you coming to sit on the LabChip. Take care, guys. Bye.
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 Bye-bye, everyone.
Thank you. Bye-bye.